Okay, let's talk about the next compound of life. So remember, those four compounds of life were carbs, lipids, proteins, and nucleic acids. Let's talk about lipids. So most of you probably know lipids is commonly referred to as fats, but there's actually three categories of lipids, and fats is only one of them. So we're going to talk about fats, of course, but then we're going to talk about things called phospholipids and steroids. And you're like, steroids? What? It's yeah. So the steroids you usually hear about in the news where athletes have been doping or whatever, those are actually synthetic forms of what nature naturally gives us. We'll talk a little bit more about that in a little bit. So whether you're talking about fats, phospholipids, or steroids, the important thing to know is that these are hydrophobic molecules. In other words, remember hydro means water and phobic means fear, afraid. So these guys are afraid of water. They're water insoluble. And this is really important because, for example, phospholipids make up the cell membrane of our cells. And if they weren't water fearing, we'd be in trouble because water would be our cells would just mix with the aqueous environment. We don't want that. So we're going to start off talking about fats, and we'll learn that the chains of fats are what we call hydrocarbons. So that just means carbons and hydrogens. So the carbons just have hydrogens all around them, and thus they are called hydrocarbons. And being that they're hydrophobic, we can also say that they are nonpolar. So you might remember this from our basic chemistry introduction. Now, unlike carbs and proteins and nucleic acids, you really can't say lipids are polymers. They don't have any true monomers to them. There are standard components, but it's not like with the other three compounds of life. So we don't really say that they're true polymers. But here are some examples of different kinds. So this would be a fat, and here you have some other fats right here and a phospholipid. So let's get into the details of these and learn about their structures and all the cool stuff that makes them lipids. So let's start talking about the structure of fats. And all fats are basically composed of the same basic structure, and that is the molecule glycerol, which kind of forms the head of the molecule, and three fatty acid chains. So I'm going to get out my pen so we can circle these things. So here is our glycerol head, our molecule, and because it's glycerol, it's polar. So even though the whole compound is considered to be hydrophobic or water-fearing and nonpolar, the head itself is actually polar, and this is going to have important properties for life. Now, connected to that glycerol molecule are three fatty acid chains. So these are your three fatty acid chains right here. And these are just hydrocarbons. They're just straight chains of carbon with hydrogen, hydrogen, joined to another carbon, hydrogen, hydrogen. So it's all the carbons are joined together, and the hydrogens kind of just surround those carbons. Notice that how are we going to actually get this fat? We're going to have to take that glycerol head and attach it to those three fatty acid tails here. So imagine this is my glycerol head. These are my three fatty acid chains. We have to attach those guys together. And so remember in carbs, the linkage was called a glycosidic linkage. In lipids, in fats, to join the glycerol head to the three fatty acid chains, we're going to form something called an ester linkage. And to do that, this is another dehydration synthesis reaction, so you should be all familiar with that by now. Notice that on the glycerol, we have a hydrogen attached to the oxygen and the carbon hydrogen right there. On the fatty acid chains, you might recognize this functional group right here. Oops, sorry, this functional group right here. Do you remember COOH? Carboxylic acid. So these fatty acid chains have a carboxylic acid at the end. And what we're going to do a dehydration synthesis reaction, we're going to take off the hydroxyl group from each carboxylic acid on the fatty acid chains, and the hydrogen off of each part of the glycerol molecule, H2O, that makes your water. And so by getting rid of water here, okay, so we're actually producing three waters, three H2Os. And apologize about my handwriting here. This little pen here, I'm using a tract on my computer to do it, so it makes it very hard to get good handwriting. Anyway, we're actually producing 3 different waters here. And so we get 3 water molecules given off for every fat that we make. And thus we have created ester linkages in each component here. And so this creates a nice fat, and because we have 3 fatty acid chains here, we're going to call this thing a triglyceride. And you may have heard this term before, triglyceride, because we have 3 fatty acid chains attached to 1 glycerol molecule, and we give off 3 water molecules as a byproduct. So that's how you form a fat, glycerol plus 3 fatty acids. Now I've put over here a picture of what adipose tissue looks like under a light microscope. Adipose is another name for fat. So if you were actually to take a sample of your fat, I got plenty of it, and look at it under a light microscope, that's what it would look like. And what's cool is inside the space is actually a oil droplets, the actual fat. And the nucleus and all the organelles are actually pushed up against the sides of the cell membrane because the fat droplet, the oil, takes up all the room. And even if you lose weight, once you've filled up that space with the oil, even if you dissolve the oil, dissolve the fat by through exercise or eating a diet, being on a diet, that space is still stretched out, and so it's very easy to fill it back up with the fat. So once you've gained weight, it's easy to gain weight again after you lose it because you're already stretched out. Pleasant thought. Okay, moving on. Okay, butter, olive oil. Which one is better for you? Well, let's look at the biochemistry to answer that question. You probably already have an idea. You've probably heard they say, oh, well, cook with olive oil, it's healthier. But let's see why this is actually. In fact, take a look. This is at room temperature. This butter is still in solid form. The olive oil is nice and liquidy. So what's going on there? Well, butter is what we call a saturated fat. And you've probably heard that before. What does that mean, that it's a saturated fat? Well, in general, saturated fats come from animals. And these actually can be what clogs your arteries. And here's why. So again, it's solid at room temperature, which means that it's easier at that kind of temperature to clog your arteries. But let's look chemically what's going on. So recall that fats are a polar glycerol head and fatty acid chain. And here is butter. And if we look at its actual chemical nature, this is stearic acid. And here's your glycerol head right here. And here's your chain. And these are all carbons. So the little black balls here represent carbon. And the little white balls represent hydrogen. So recall that each carbon is going to have 2 hydrogens attached to it. Now notice that all of these bonds here are single bonds, and each carbon is joined to a carbon on one side and a carbon on another, and then 2 hydrogens. So in other words, each carbon is saturated with hydrogens, hence the term saturated fat. And because of that structure, it causes the fatty acid chain, the hydrocarbon chain, to be straight, which causes butter to be solid at room temperature. And so if you think about what's going on in your arteries, when you're consuming lots of animal fat over time, that is building up plaque inside your arteries, causing atherosclerosis, which can contribute to heart disease. So not good, not good. But again, saturated fats, carbons are completely saturated with hydrogens. We contrast that to an unsaturated fat like olive oil, fish oil, all those kinds of things. These are much better for you. They are, because of the chemical structure, they are liquid at room temperature. And so if we take a look at this, so this happens to be canola oil, which I actually don't recommend consuming. That's a whole other discussion. But any unsaturated fat, whether it's olive oil, canola oil, fish oil, whatever, it looks like this chemically. So you have your polar glycerol head, and here is your carbon hydrogen chain, your hydrocarbons, but something's a little bit different here. If you take 2 of the carbons, you actually could have more than that, but at least 2 of the carbons, and notice that each carbon, in this particular case right here, only has 1 hydrogen bonded to it. And instead of having two hydrogens, it has one hydrogen and a double bond between the two carbons. And so that double bond, recall that double bonds are shorter than single bonds and stronger. And thus, instead of it being a straight chain, it kinks up at that double bond. And so this kink is what's responsible for keeping that unsaturated fat, because the carbons are unsaturated, they're not saturated with hydrogens. It causes it to remain liquid at room temperature, flows much nicer through your arteries. So a little bit better for you than saturated fats. I should make a caveat though. It depends. This is in their natural form. We humans have gotten all Frankenstein-y and decided to alter unsaturated fats in the laboratory in order to give them longer shelf life. And this is actually quite bad for our health. So normally, if you took an unsaturated fat like olive oil or like this canola oil here, notice that the hydrogens at the double bond, so the hydrogens that are attached to the carbon here, are both on the same side. Moose configuration, right? Like moose antlers, they're on the same side. A Latin prefix that means same is cis. So those hydrogens are cis configuration. They're on the same side as each other. And so we can take a look at this particular unsaturated fat, oleic acid, and notice again, here's your double bond where the kink in the chain is, and the hydrogens are on the same side. So it's the cis configuration. Bodies are okay with that. We can handle that. But you can chemically change the configuration of those hydrogens around the double bond so that they're doing the John Travolta thing, remember? Where one hydrogen's on one side and the other one's on this side. So this is called the trans configuration. They are on the opposite sides of each other. So this is a trans fatty acid. And trans fats, you've probably heard, are really bad for you. That is one of the biggest contributors to heart disease in this country. And so there's definitely a big movement to try to eliminate trans fats from the American diet. So generally, unsaturated fats are better for your health than saturated, but not if they're in the trans configuration. Fun stuff to tell your friends at parties. Okay. So I think we have a good understanding of fats. Now let's move on to phospholipids. Phospholipids are so important to life because, again, these are the major component of our cell membranes. And that's true whether you are any kind of animal, like a human, like a duck, like a platypus, or any kind of plant, or any kind of protist, or even bacteria. Our cell membranes contain what's called a bilayer, 2 layers of phospholipids. So these, you'll recognize the majority of the components of these just from your understanding of fats. They have fatty acids, although their fatty acid chains, they don't have 3, they have 2. So they have 2 fatty acid chains attached to a glycerol by ester linkage, just like you learned before. But then they got this head right here. And this is called a phosphate head. It is polar. So you'll remember phosphate groups from your study of functional groups, hopefully. And because phosphate has such a large negative charge, 3-, that makes that very, very polar. And so you have this strong polar head attached to polar glycerol and then 2 nonpolar fatty acid chains. So it makes for very interesting properties of this molecule. Now, in the case of our cell membranes, and we'll get into this a lot more detail when we study cell membranes in detail, you'll notice that our cell membranes have 2 layers, a bilayer. Bi means 2. So 2 layers of phospholipids. And because of the chemical properties of this and the aqueous environment that they're in, you get them kind of going in opposite directions. So imagine my wrist was the phosphate head, and I have my 2 fatty acid chains here, which are hydrophobic. You'll end up getting a configuration like this, where one has the polar head facing the external environment, and the other one has the polar head facing the inside of the cell. And so that's how this thing forms the cell membrane that goes all around the cell. So more about that later. But notice here that one of the chains has a kink and the other one doesn't. So in the case of phospholipids that make up our cell membranes, one is a saturated fatty acid chain where it's straight and all single bonds, and the other one has a double bond right here And well, I did not draw that very straight. There we go. Double bond right there, which causes one of the tails to be kinked. So one is straight and one is kinked like that. So it's really cool. You can take phospholipids and put them in water, and they will self-assemble into a bilayer like this. Even if it's not an actual cell, you can make what are called protocells, sort of cell-like things. It's pretty cool. So just studying the properties of phospholipids and how they self-assemble in water gives us some insight into a probable way that the first cells arose during evolutionary time. So here's a deeper look at the structure of a phospholipid. So note here, you have your phosphate head, your glycerol backbone. And so this right here makes your polar head group. This makes your glycerol, and then here are your two fatty acid chains. This particular one is not showing a double bond in one of them. The other one is all straight as well. But remember that for the phospholipids making up our cell membranes, one is kinked, one's unsaturated, and the other one's saturated. Okay, and our final group to talk about, steroids. So cool. So it may surprise you some of the things that are categorized as steroids. So when you hear about anabolic steroids, the synthetic ones that people take illegally, you know, athletes, for example, those are actually synthetic forms of the steroid testosterone. And you're like, testosterone, right? The male hormone. And actually females and males both produce testosterone. It's just males tend to have more cell receptors on their cell membranes that respond to the testosterone than females do, and they also produce more. But in general, steroids are a category of lipids that are used for cell signaling. So yes, our trillions of cells in our body communicate with one another, and they do it chemically, and they can do this through the use of steroids. So we'll be getting into a lot more detail about cell signaling, cell communication later. But for now, let's concentrate on the structure of steroids and some other cool things. So, so these guys can serve as hormones. And it may surprise you that not only testosterone and estrogen, the sex hormones for males and females respectively, not only those are hormones, but check this out, cholesterol. Yeah, the stuff that's in our butter. Remember butter? Butter. Better with butter. Yeah, cholesterol is a steroid, and in fact, cholesterol is the precursor compound to testosterone and subsequently to estrogen. So let's take a look at this. So one thing that these steroids have in common is they have a 4-ring structure as their backbone. So you get 4 rings stuck together, and that's what gives the steroid its properties. And the only difference from one steroid to another are those functional groups that are attached to it. So again, hopefully you're studying your functional groups because it'll give you a much better understanding of what's going on here. So let's look at cholesterol. So notice we have a 6-carbon ring and a 6-carbon ring and a 6-carbon ring and a 5-carbon ring. And so this is the basic 4-fused carbon ring backbone of steroids. Now in the case of cholesterol, notice we have a hydroxyl group stuck here, a methyl group there, a methyl group there, and then this big chain. Don't forget everywhere where there's a little corner, that means there's a carbon. So that's cholesterol. That is the chemical structure of cholesterol. Now let's just chemically alter this a little bit. Let's break some bonds and change some functional groups around. So let's take off this great big chain right here and replace it with a hydroxyl group and break a bond over here to make this a double bond to the oxygen. Lo and behold, you have just gone from cholesterol to testosterone. Things that make you go, And if you alter that a little bit further, so let's go from testosterone, and let's go ahead and keep our hydroxyl group and our methyl group there. Let's take off this methyl group, and let's go back to our hydroxyl group here. So we're going to break this double bond, go back to a hydroxyl. You just went from testosterone to estrogen. Isn't that cool? So this is what our bodies do. Remember, we don't like to reinvent the wheel. So we're just going to take one compound, alter it, often with the help of some enzymes to catalyze the reactions, and go from one compound to another. So testosterone is a precursor to estrogen, and cholesterol is a precursor to testosterone. How cool is that? So imagine in the evolution of life on Earth, the evolution of more complex life forms, slight chemical changes make big changes when it comes down to it. How cool is that? Any questions about lipids? Let me know. We still got proteins and nucleic acids to go